Imagine this, a brand new medical device, maybe a revolutionary diagnostic tool, or perhaps a life-saving drug that promises to transform patient care. You're holding it, or you're about to receive it. How do you truly, fundamentally know it's safe? How can you be absolutely certain that the incredibly sophisticated machines that manufactured it are reliable, consistent, and won't introduce any harmful contamination into something so critical? The stakes, well, for all of us, they really couldn't be higher. Okay, let's unpack this complex yet absolutely essential world of validation. It's sort of the invisible shield that ensures the safety and efficacy of everything from your everyday over-the-counter medication to cutting-edge advanced therapeutics. This isn't just about, good practice. It's a meticulously documented journey from a blue sky design all the way to a perfectly safe and sterile product. It's far more intricate and, frankly, far more fascinating than most people probably realize.
What's truly fascinating here, I think, is that we're looking beyond just a single piece of equipment or a solitary step. We're connecting the dots across an entire manufacturing ecosystem, understanding how each individual step contributes to the ultimate unwavering goal of consistent product quality and, crucially, patient safety. This raises an important question for all of us. How do we build verifiable, ironclad confidence in such intricate systems, especially when they're producing something so critical, something that literally holds lives in its balance?
Absolutely. Building that confidence is paramount. It has to be. Today, we're embarking on a deep dive into the world of equipment qualification and process validation within FDA-regulated industries. We're going to demystify what can seem like an alphabet soup of acronyms: IQ, OQ, PQ, FAT, SAT, and the ultimate crucible test, media fills. But these aren't just bureaucratic hurdles. Think of them as the meticulous and visible guardians of public health, rigorously ensuring that what's inside that vial, that syringe, or that medical device is exactly what it's supposed to be, without compromise every single time. To guide us through this intricate landscape, we've gathered insights from comprehensive industry guides, candid expert interviews, and some real-world case studies of incredibly complex sterile filling lines. Our mission today is to demystify these critical processes, to illuminate why they are utterly essential, how they are executed with such precision, and the significant challenges companies navigate to ensure every single step meets the highest possible standards. You're going to walk away with, hopefully, a crystal clear understanding of the monumental effort it takes to bring safe, effective products to market. So let's start at the very beginning with the foundational concept of qualification. When we talk about qualifying equipment in the pharmaceutical or medical device world, it sounds a bit like, getting a certificate of approval. But in pharma, it's so much more profound than just a piece of paper, isn't it? It's about absolutely proving, meticulously documenting, and consistently ensuring that every single piece of equipment and every utility, from a complex processing machine to the air handling system, is perfectly suited for its specific intended job and operates optimally. And here's the kicker. This entire process has to happen before any commercial scale manufacturing even begins. It's the absolute prerequisite, like the birth certificate of your manufacturing capability.
Precisely. Qualification is a systematic, documented process that goes far beyond a simple checklist. It confirms that the equipment itself, all associated piping, instruments, and utilities, everything from compressed air and water for injection, WFI, to purified steam, are not only installed correctly, but also operate exactly as intended. And critically, they have to perform consistently according to their detailed design specifications. The goal here is to create an undeniable, detailed, documented evidence trail. This trail often requires multiple signatures from key departments like validation, engineering, maintenance, calibration, and quality assurance. This isn't just for internal use, it's a verifiable, auditable record for regulatory agencies and clients, proving that this critical equipment has been delivered, installed, and configured precisely as per engineering drawings and design specifications. It's truly about building that foundational trust, piece by painstakingly documented piece.
And this isn't just some optional good practice. We're talking about a legal imperative, right? Yeah. A regulatory demand for this level of rigor. For example, the FDA's 21 CFR 211.63 explicitly states that equipment used in manufacturing drug products must be of appropriate design, adequate size, and suitably located to facilitate operations for its intended use and for its cleaning and maintenance. No, it's not a suggestion. It's a non-negotiable requirement.
And if we connect this to the bigger picture, the why behind this legal mandate is entirely about mitigating risk. Think about it, even a slight installation error, or a seemingly trivial performance problem with a valve or a sensor could cascade into a serious, even catastrophic product quality issue. This risk is amplified with novel systems that don't have a long history of performance to rely on. A minor miscalibration or a faulty connection identified late could lead to contamination, incorrect dosing, or compromised sterility. In industries where products are literally going into or onto people, these issues can have deadly consequences for patients. So, this isn't just about ticking boxes for compliance with FDA regulations or following guides like EU GMP Annex 15 or the ISPE Baseline Guide 5, which are indeed widely adopted and supported by regulatory authorities. It is fundamentally about preventing potentially catastrophic outcomes and saving lives. The monumental upfront investment in qualification, well, it pays dividends in patient safety and avoided crises down the line.
To help visualize this incredibly intricate process, the industry often uses something called the V model. It sounds a bit technical, but it's actually a very intuitive visualization tool, isn't it? Think of it as mapping the entire life cycle. On the left side of the V are all the planning steps from your initial user requirements and the functional design to the detailed engineering blueprints. Then, the right side of the V links those directly to the execution stages of Installation Qualification, IQ, Operational Qualification, OQ, and Performance Qualification, PQ. It beautifully illustrates how every planning step on the left has a corresponding, verifiable execution and reporting step on the right, all built upon a solid risk-based approach. It's a complete lifecycle view, ensuring that hopefully no requirement or potential risk is overlooked. So even before that shiny new, incredibly complex piece of equipment even arrives at a pharmaceutical manufacturing plant, there are crucial testing phases that occur to set the stage for success. This isn't just about getting a delivery, it's about rigorous checks at multiple points, mitigating risk long before installation.
Absolutely. These pre-installation tests, known as factory acceptance tests, or FAT, and site acceptance tests, SAT, are pivotal in the overall validation lifecycle. They're typically outlined right in the validation master plan, acting as proactive checkpoints. The real insight here isn't just what they test, but that catching an issue at FAT might cost, I don't know, a few $1,000s in a few days to resolve. Fixing that same issue once the line is installed, validated, and potentially running commercial batches could easily be in the millions. Not to mention months of production delays. It's truly a monumental upfront investment that proactively saves immense effort, time, and costs downstream. It just makes sense.
Let's start with the Factory Acceptance Test, or FAT. As the name suggests, this is where the magic begins at the system manufacturer site. It's a series of tests conducted by the vendor to verify they've met all their responsibilities, everything from regulatory compliance to the client-specific user requirements. The purpose is pretty clear: to convince customers, including the client's technical and quality teams, that they're delivering high-quality equipment and to ensure every component and control functions exactly as specified. Crucially, it's about catching any issues early on before the equipment is even shipped across continents, allowing them to evaluate the equipment thoroughly against its design specifications in a controlled environment. So when the validation and engineering teams are at the manufacturer site for an FAT, what are some of the absolute non-negotiables they're meticulously checking for? What really stands out as critical here?
You're right to highlight the critical checks. While the full list can be extensive, some of the most impactful activities during an FAT include: first, drawings and documentation verification. This means meticulously checking all the detailed blueprints, including the PNID drawings, that's piping and instrumentation diagrams, which are essentially the detailed schematics of the system's plumbing and instrumentation to ensure they match the physical equipment. Second, critical instrumentation calibration verification is essential, confirming that all sensors and measuring devices are accurate before they leave the factory. You have to know they work. And third, they'll perform comprehensive software and hardware verification, checking everything from input-output functions to alarms, interlocks, access levels, and recipe management systems. It's about ensuring the brain and nervous system of the machine are fully functional and precise from the get-go. We see excellent examples of this collaborative approach in practice. Vern Stroder, the CEO of Culinard, for instance, described how they invite clients like Berkshire Sterile Manufacturing, or BSM, to their site to personally witness test runs for various container types and sizes. During this, they're generating a full qualification documentation package, including the detailed FAT protocols, which is then refined in real-time collaboration with the client. It's very hands-on. Similarly, Genesis Packaging Technologies provides extensive documentation to BSM before the FAT even happens, supporting BSM's own pre-validation efforts. And then they collaboratively refine that documentation during the FAT execution at their facility. This upfront collaboration is absolutely critical for smooth downstream validation. It prevents so many headaches later.
Okay, so once that equipment successfully passes the FAT, it's packed up and ready to travel to its new home. That's where the Site Acceptance Test, or SAT, comes into play. This series of tests is conducted not at the manufacturer's, but at the owner representative site, meaning the actual pharmaceutical plant where it will be installed. Its primary critical purpose is to provide documented evidence that the equipment was delivered in pristine condition and wasn't adversely affected by the rigors of transportation. Like, did it survive the journey? It essentially builds upon the FAT efforts, confirming everything still works as expected post-transit.
The key activities for an SAT are certainly similar to FAT in scope, but are laser-focused on on-site verification. This typically includes confirming critical instrumentation calibration again, performing input-output checks, a visual verification of P&ID drawings, and thorough startup procedures. There's also the crucial punch list, where any final discrepancies or minor issues found upon arrival, perhaps a dent from shipping or a missing cable, are meticulously documented and addressed before full installation proceeds. Got to catch those little things.
So what's the fundamental defining difference between FAT and SAT for our listeners, just to make it really clear?
The most obvious, of course, is location. FAT is at the manufacturer's site and SAT is at the owner's. Simple enough. But a critical and often underestimated distinction lies in the environmental conditions. At FAT, the equipment might be tested with simulated utilities and ambient conditions. However, at SAT, the equipment is connected to and uses real site utilities, things like compressed air, high purity water for injection, WFI, and plant steam. And it experiences the actual environmental conditions, such as the specific temperature and humidity of the manufacturing plant's cleanroom. This is vital because real-world variables can expose issues that simulated conditions might miss. Things behave differently sometimes. The documentation also differs. FAT protocols are extremely detailed, verifying every design aspect, while SAT is generally simpler, focusing on verifying successful delivery, initial functionality, and readiness for installation.
This brings us to a crucial, efficiency-driven concept in modern validation. Leveraging strategy. It sounds like a smart way to streamline things. How does it work?
It absolutely is a smart way. As per ISPE guidelines, leveraging allows a company to utilize properly documented activities and data from FAT and commissioning those initial startup tests to directly support the subsequent IQ and OQ phases. The core benefit is avoiding unnecessary repetitions of tests, which can significantly reduce the overall qualification time and cost. Saves time, saves money. However, this strategy isn't a shortcut. It demands exceptionally good documentation practices from the vendor, a predefined scope and depth of testing that's mutually agreed upon with the client, and clear agreement on how raw data will be collected and presented. You need that rigor. Stephen Kinney, BSM's Senior Validation Manager, confirmed that BSM embraces this approach. They perform SAT with supplier assistance to ensure equipment is working correctly after shipping, and their subsequent IQ/OQ will leverage much of the testing already performed within the SAT. It's a pragmatic approach to streamlining a complex, resource-intensive process without compromising quality. Work smarter, not harder, right?
With the equipment now delivered, checked at the site, and those critical pre-installation hurdles cleared, we embark on the truly foundational phase of qualification. This is where IQ, OQ, and PQ truly take center stage, each building upon the last, like constructing a meticulously engineered building floor by floor. Hashtag 3.1 Installation Qualification, IQ. Let's start with Installation Qualification, or IQ. After all the equipment has arrived, its purpose here is to meticulously verify that every single piece of equipment, all associated piping, software, and instruments are correctly delivered, installed, and configured. This must precisely match the design specifications and the manufacturer's installation checklists. Essentially, IQ ensures the system has the necessary prerequisite conditions to function as expected. It's about meticulously checking every physical connection, every wire, and every software setting, getting the physical setup right.
You've hit on the essence. The key focus areas for IQ are incredibly thorough because it's the birth certificate of your system, as you said. This includes verifying the physical location and necessary floor space, documenting all computer-controlled instrumentation, verifying all manuals and certifications, and meticulously unpacking and cross-checking instruments for any shipping damage. Did it arrive okay? We're also talking about ensuring the correct power supply is in place, installing all ancillary instruments, and critically documenting specific firmware versions and serial numbers. Missing a firmware version now is like forgetting a detail in that birth certificate. It can cause legal and operational nightmares years later. Seriously, it happens. It also covers verifying environmental and operating conditions, such as ensuring a clean room maintains its specific temperature range, as well as the correct software system installation, accessibility, and robust connections with peripheral units. Finally, recording the calibration and validation dates of any tools used during the IQ process is essential. That traceability is key.
What then truly makes an IQ successful? What's the ultimate indicator that this crucial first step has been done correctly?
What makes an IQ truly successful is its ability to directly compare what was specified in the design documents against what was found on site during installation. And meticulously recording any noncompliance or deviations, it's about that direct, documented comparison, demonstrating that the physical reality precisely matches the engineered design, is what you got, what you asked for. And beyond these basic checks, there are critical best practices for writing IQ protocols that elevate them from mere checklists to powerful risk mitigation tools. Integrating risk management from the start, for instance, means prioritizing checks on things like critical software versions or configuration settings based on their potential impact on product quality, where a tiny error could have huge consequences. Drawing on historical data from similar equipment can reveal common installation challenges or calibration drift issues, allowing for proactive adjustments rather than reactive fixes later on. Learning from the past. Specifying clear, measurable acceptance criteria is also vital. Instead of a vague install as per manufacturer's instructions, an IQ protocol should specify, for example, ensure the centrifuge rotor speed reaches 15,000 RPM and 900 RPM as per manufacturer's operational specification, making it quantifiable. No ambiguity. Furthermore, robust protocols clearly cross-reference related documents like the validation master plan or design qualification, creating a cohesive, auditable documentation suite. Everything connects. A predefined process for handling nonconformities, how they're documented, evaluated, and resolved, is essential for transparency and continuous improvement. What do you do when things go wrong? Thinking ahead to future flexibility for upgrades, using visual aids like diagrams and photos for complex wiring, trust me, photos help a lot, and planning for environmental considerations like required temperature and humidity controls are also crucial. And of course, ensuring that all personnel executing the IQ protocol are thoroughly trained, not just on the equipment, but on the critical importance of accurate documentation for regulatory compliance. The people matter.
The documentation itself for IQ is extensive, encompassing the IQ protocol, which details the scope, methodology, and criteria, a detailed IQ checklist for step-by-step verification, and the comprehensive IQ report, which summarizes all findings, observations, and results. Paperwork, 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 but essential paperwork. For Berkshire Sterile Manufacturing, Stephen Kinney explains that for their sterile filling line, IQ ensures crucial elements like power requirements are met. Proper hookups for critical utilities like WFI, that's water for injection water, and drain systems for the vial washer are in order. It also involves reviewing relevant documentation, the engineering turnover package, and confirming the readiness of procedures, calibration maintenance schedules, and equipment activation packages. It's a truly comprehensive first step that leaves no stone unturned. Hashtag tag 3.2 operational qualification. OQ. Okay, once that meticulous installation qualification is complete and everything is physically in place and connected correctly, we then move on to operational qualification, or OQ. This is the phase where we determine that the equipment's performance is entirely consistent with the user requirement specifications, and that it reliably operates within the manufacturer's specified ranges. It's where we identify and meticulously inspect features that directly impact the final product quality. Does it actually work as intended? And remember, this only happens after a successful IQ. You can't skip ahead.
That's right. The key focus areas for OQ often delve into specifics that directly impact process control and product quality. This includes things like precise temperature control and variations within critical zones, the functionality of servo motors and air flaps controlling delicate movements, temperature protection systems, and even security aspects like card readers and access systems. Can only the right people use it? It also rigorously verifies pressure and vacuum controllers, CO2 controls for maintaining specific atmospheric conditions, humidity measuring and control systems, fan speeds, and the proper functioning of display units and signaling LEDs. Does it light up when it's supposed to? Essentially, every operational aspect that could influence the quality, safety, or consistency of the product is rigorously put to the test.
So after putting the equipment through its paces, what signals a truly successful OQ? What are the key indicators that it's ready for the next stage?
A successful OQ means several things. First, all operational tests meet or exceed the predefined acceptance criteria in the protocol. Passes the tests. Second, there should be no significant deviations during testing. Nothing weird happened. Third, all error detection and handling mechanisms need to function exactly as expected. The machine must correctly identify and respond to faults. Does it know when something's wrong? If the equipment integrates with other systems, that integration needs to be seamless and verified. Does it play well with others? And crucially, positive operator feedback indicates the equipment is user-friendly, effective, and reliable in day-to-day operations. Do the people using it think it works well? Ultimately, a successful OQ means the equipment is robustly proven to operate as intended and is fully ready for the next critical phase, performance qualification. Ready for the big show. For OQ protocols, best practices extend significantly beyond mere functionality checks. It's absolutely vital to test the full operating range, not just nominal conditions. This means pushing the upper and lower limits, for example, operating a tablet press at its slowest and highest speeds to ensure consistent tablet weight and hardness across the entire spectrum. Don't just test the easy middle ground. It's also critical to simulate failure modes to verify error detection. For instance, intentionally creating scenarios where a LIMS, that's the Laboratory Information Management System, might receive corrupted data to confirm its integrity checks and alarms function correctly. Try to break it, basically. Testing interoperability with other systems is also key. Does an analytical instrument seamlessly transmit data to the LIMS, and is that data parsed and stored accurately? Does the data flow? Furthermore, OQ should include testing under different environmental conditions where applicable to assess reliability across various production scenarios. What if it's warmer or more humid one day? Data integrity and accuracy verification, including rigorous access control for different user roles within the software, is non-negotiable. Can someone tamper with the data? No. Good. Sequential operation testing, mimicking actual production cycles, ensures components work in harmony. Does it work repeatedly? And proper load testing, from minimum to maximum capacity and varying loads, confirms performance consistency under the most demanding real-world production demands. Can it handle the pressure? This comprehensive approach ensures robustness and uncovers hidden vulnerabilities before they impact patients.
The documentation for OQ is equally comprehensive, including the OQ protocol, OQ test script checklist, the OQ report, and all relevant standard operating procedures, SOPs, calibration, and maintenance records. More paper. A traceability matrix is also crucial, linking the design specifications and user requirements directly to the OQ test, ensuring nothing is missed and that every requirement is demonstrably met. Connecting the dots. Stephen Kinney from BSM highlights that their OQ tests the equipment's operations to ensure all operating specifications and user requirements are met. For their filler, for instance, a requirement is to fill vials from 2R. Those are small vials to 100R sizes, which are much larger. So the OQ rigorously documents successful filling of both extremes. And importantly, BSM writes and performs all OQs for their line with support from the equipment supplier, ensuring direct ownership and expertise. They're driving the process. To really bring these OQ concepts to life, let's look at some fascinating real-world examples from the suppliers building BSM's cutting-edge line. You'll see how diverse and specific these tests can get.
These examples truly illustrate the depth of OQ. Take Steriline's Vial washing machine OQ. The main test is its cleaning capacity. Vials are intentionally soiled with a riboflavin solution that's a fluorescent compound often used to simulate tough visual contamination, then run through the machine, and must emerge perfectly clean. Can you see any residue? Nope. Okay. It's a direct, visible, and quantitative measure of effectiveness, ensuring no residues remain. Then there's Steriline's pyrogenating tunnel OQ, which is all about verifying the proper function of its hot and cooling chambers. Hot enough to kill bugs, cool enough not to wreck the product. Thermocouples are placed inside vials in worst-case positions. Imagine a vial completely surrounded by others, with a thermocouple at the very bottom, the hardest place to heat. This ensures the critical temperature for depyrogenation, achieving a 6-log reduction in pyrogens, meaning a million-fold decrease in those fever-inducing substances is reached and maintained. And then that they cool down to a suitable temperature for filling without damaging the product. Critical control. Another fantastic example comes from Culinard's flexible filler OQ. Bernstrader details how they test the most challenging container size first, for example, running 100 of 4R vials, filling at least 500 units without issue. Start with the hard stuff. Once that's proven, they move to other container sizes, filling maybe 1-2 trays, 5,200 units without issue, repeating this for syringes and cartridges too. Since sophisticated robots are integral for loading and unloading, their precise, issue-free performance is also verified. Do the robots drop anything? Hopefully not. And for Culinard's new 100% in-process weight check technology, which measures dispense volume for each container in real time, well, its OQ is rigorous. They fill a specific number of containers, record the real-time volume, and then perform a traditional, highly accurate weight check on a separate scale. Compare the new tech to the old standard. By using the known density of the solution, they can accurately verify the precision of their real-time measurements, ensuring critical dosing accuracy for every single unit. That's huge for safety.
And what about Genesis Packaging Technologies, especially with their capping systems, vision systems? How do they validate something that's essentially seeing potential problems? That sounds tricky.
Genesis has some unique and robust validation methods for their capping systems, vision systems OQ. It's pretty cool, actually. For the raised stopper inspection, which happens pre-capping, they validate that the system correctly identifies raised, tilted, or missing stoppers, or even missing vials, ensuring no improperly sealed products move forward. Catching errors early. For the post-cap inspection, they confirm it accurately identifies missing plastic cap buttons or missing caps, which are critical for product integrity and pamper evidence. The validation method involves running specific numbers of good packages and verifying incoming and passing vial counters. Count the good ones. Crucially, they also run a mix of good and bad packages, verifying the counts and, most importantly, ensuring the automatic machine shutdown functions correctly when excessive consecutive failures are detected. If it sees too many bad ones in a row, it stops. That vital control prevents widespread errors and ensures continuous quality. Steriline's robotic tub decontamination system also undergoes a specialized OQ. They verify light uniformity using a joulemeter, a device that measures light energy ensuring pulse sleep intensity is uniform and energy is above 1 joule within a specified min-max range. Is the bug zapping light even? The decontamination effect itself is verified by placing biological indicators, tiny controlled populations of resistant microbes in worst-case points, such as under the Tyvek cover, that's a breathable, sterile barrier, or below the tub border edges to ensure a 4-log reduction. Did the light kill the tough bugs? And finally, Steriline Supervisory PCOQ focuses on software functionality, rigorously testing the audit trail who did what when, batch creation export, user permissions, trend analysis, line output, can it run for 30 min straight per format, and the pressure cascade system. That pressure cascade maintains precise differential pressures in different cleanroom zones to keep contaminants out. All that needs testing across various operating modes. It's a testament to the depth of testing required for every single component, whether mechanical, optical, or digital. #Trick 3.3 Performance Qualification, PQ.
All right, so after all that meticulous installation and operational qualification, where every component and its function has been rigorously tested, we arrive at Performance Qualification, or PQ. This is the final and perhaps most critical step in equipment qualification. It's where the team verifies and documents that all user requirements are consistently met, ultimately confirming that the equipment and entire systems are truly fit for intended use, as defined in the user requirement specification. The final proof. This only proceeds after successful IQ and OQ. No shortcuts.
The key difference from OQ is significant and crucial. While OQ tests individual components and their operational limits, PQ tests them all as a partial or overall process. Under actual production conditions, and crucially with product or simulated product, underload, real conditions, real stress, this means the tests cover the full operating range with the equipment experiencing real-world stress, simulating batch production to identify any faults or inconsistencies that only emerge when all individual components are interacting harmoniously as a complete system. Does it all work together smoothly?
The FDA's guidance on process validation, in fact, defines the PQ stage in 2 elements. First, the design of the facility and qualification of equipment utilities, and second, the process performance qualification, PPQ. They make it very clear that current good manufacturing practice, CGMP compliant procedures, must be followed, and successful completion of Stage 2, PPQ, is absolutely necessary before commercial distribution of the product can begin. It truly is the final gate before launch.
The recommended elements for PQ and PPQ protocols are extensive and comprehensive, designed to ensure product quality and consistency over time. They include clearly defined manufacturing conditions, such as equipment limits and operating parameters, a thorough list of data to be recorded and analyzed, and specific tests designed to ensure consistent quality throughout production runs. A detailed sampling plan, robust analysis methodologies, often statistical and risk-oriented, clear definitions for variability limits, and contingency plans for non-conformance are also vital. What's acceptable variation? What if it goes outside that? And of course, the quality unit must review and approve the entire PPQ protocol, underscoring its immense importance and their ultimate responsibility. Quality has the final say.
So beyond the standard checks, what are some of the cutting-edge or best practices for PQ and PPQ protocols that truly push the boundaries of quality assurance? What's the state-of-the-art thinking here?
This is where real innovation comes into play. Best practices for writing these protocols suggest incorporating real-time monitoring tools, like inline spectrophotometers for continuous tablet coding uniformity checks. This allows for immediate feedback and adjustments, rather than waiting for post-production quality control hours or days later. Catch problems as they happen. Another powerful approach is applying process analytical technology, or PATE tools, which can provide real-time feedback and allow for dynamic adjustments. Think near-infrared NIR spectroscopy to monitor moisture content during granulation. These tools are revolutionary because they shift from reactive quality control to proactive process understanding and control. Know your process, control your process. And critically, protocols should be designed for scalability, evaluating process capacity limits. For instance, if current demand is for 100 units per batch, conducting PQ runs at 150-200 units tests the process's ability to scale up without compromising quality, proactively addressing future growth. Can you make more if needed?
The ultimate output of this phase is, of course, the comprehensive PQ report, formally confirming that the machine meets all performance specifications. The final sign-off. Stephen Kinney from BSM explains that after individual IQOQs pass, BSM doesn't stop there. They develop a cohesive process through PQ. This ensures reproducible results within a working range under real-world conditions. Can we do it again and again reliably? Individual PQs for specific equipment, like the pyrogenation tunnel, vial washer, and VHP, this vaporized hydrogen peroxide system, are performed, often with associated recipes or operating parameters. But the real culmination, the true measure of readiness, is a final PQ on the entire line as a whole. This signifies the complete readiness of the integrated system to reliably produce quality products, the whole orchestra playing in tune. Here's where it gets really interesting and, frankly, incredibly challenging. Imagine building a cutting-edge sterile filling line, a true marvel of engineering, with technology sourced from not just one, but multiple highly specialized suppliers from around the globe. How do you possibly validate something so intricate, where every single piece has to talk seamlessly to every other piece, and the entire integrated system must operate as one harmonious, sterile unit? This is the very real and demanding reality for Berkshire Sterile Manufacturing, or BSM, with their new state-of-the-art line. It sounds like a validation nightmare.
Indeed, BSM's new line incorporates a truly diverse array of advanced technologies. We're talking about the high-precision filling system from Culinard, a sophisticated capper from Genesis, a powerful lyophilizer from Ime, and then from Steriline, an entire integrated isolator system, all the critical transport mechanisms, container processing equipment like the vial washer and pyrogenating tunnel, the lid liner system, a robotic tub decontamination system, and the overarching supervisory PC. Wow, that's a list. What makes this particularly noteworthy and adds an extra layer of complexity is that some of this technology is genuinely brand new in the fill-finish world, like the innovative RTU ready-to-use tub decontamination chamber and Culinard's 100% in-process weight check technology. New stuff always adds risk.
This isn't just a collection of machines. It's a fully automated, isolator-based, highly flexible line designed to handle an enormous range of products. Bulk vials, RTU vials, syringes, cartridges, lyophilized products, and even highly viscous solutions. It's a true manufacturing workhorse. Stephen Kinney, BSM's senior validation manager, freely acknowledges the sheer scale of this undertaking, stating that the validation process alone is expected to take at least 6 months from the factory acceptance test all the way through to performance qualification. 6 months. That's a monumental task requiring incredible coordination.
And as we've already detailed through those IQOQ examples, each vendor's specific validation contributions are absolutely crucial to the whole. Culinard's flexible filler and its 100% in-process weight check, Steriline's comprehensive isolator and processing equipment like that pyrogenating tunnel, ensuring a 6-log pyrogen reduction, and the robotic tub decontamination along with Genesis's capping system with its sophisticated vision systems, all bring their unique, rigorously validated elements. The success, integrity, and regulatory acceptance of the entire line ultimately rely on the individual strengths of its meticulously qualified parts and how flawlessly they integrate. The chain is only as strong as its weakest link, right?
In such an incredibly complex multi-vendor environment with so many moving parts and brand new technologies, quality assurance plays an absolutely pivotal, almost orchestral role. It's not just an oversight function. It's intrinsically integrated into every single step. They're not just checking at the end.
This truly highlights that quality isn't just a department, it's an integrated philosophy that permeates the entire project, from initial concept to final product. Debbie Smith, BSM's VP of Quality Assurance, emphasizes that QA's role involves comprehensive oversight for all aspects of the validation plan. They're everywhere. This means rigorously reviewing and approving all pre-executed protocols for FAT, SAT, IQ, OQ for individual equipment, and PQ for the entire line. They read everything. Then, after execution, QA meticulously reviews these protocols again to ensure documented evidence of proper installation, adequate support utilities, verification of overall operating ranges, and acceptable performance. Did it actually meet the plan? This critical step ensures that the equipment meets all specifications, its intended purpose, and crucial regulatory requirements like CGMP, FDA, and EU guidelines, ultimately allowing it to be released for GMP good manufacturing practice manufacture. The final green light. Furthermore, if any discrepancies or unexpected events, what we call deviations, occur during testing, QA collaborates closely with engineers to ensure thorough documentation. This means detailing the occurrence, meticulously identifying the root cause, outlining effective corrective actions, and assessing the impact on the system or product. What went wrong? Why? What did we do? And did it affect anything else? Any resulting changes to the equipment or process must not, under any circumstances, compromise the established requirements or validated state. Fixes can't break something else. This constant vigilance, from protocol approval to real-time observation and expert deviation management, is absolutely critical to building that verifiable trust in the entire product realization process. It truly is the ultimate guardian of patient safety, ensuring a product is safe not just by chance, but by design and documented proof.
So we've meticulously qualified all the individual pieces of equipment. We've seen how they flawlessly come together in a complex multi-vendor line. But now it's time to validate not just the equipment, but the process itself. And this is where we get to the ultimate test, the crucible of sterile manufacturing, aseptic process simulation, universally referred to as media fills. As Stephen Kinney from BSM mentioned, once those individual equipment PQs pass, BSM then performs a final PQ on the entire line as a whole, which is precisely a series of these media fills. The final exam.
Aseptic process simulation, or APS, is an incredibly critical tool used to evaluate the capability of the entire aseptic processing activity, the ability to keep things sterile. Can you really make a sterile product? It does this by using microbiological growth-promoting media, such as soybean casein digest medium, SCDM, for aerobic microorganisms, or fluid thioglycolate medium for anaerobes, in place of the actual product. This media acts like a magnet for any microbes. If there's a bug anywhere, it should grow in this stuff. The simulation meticulously covers the entire aseptic process, from the initial product and component sterilization all the way through to the final sealing of the container. Every step the real product sees. The media contacts all product contact surfaces of the equipment chain, the container closure, the critical cleanroom environment, and every single process manipulation that the actual product will undergo.
So what's the driving purpose behind such an elaborate organism feeding test? Why go to all this trouble with media instead of the actual drug product? It seems like a lot of effort.
The purpose of these media fills is multifaceted and absolutely vital. First, it assesses the manufacturing line's capability to consistently and reproducibly produce sterile products. Can you do it over and over? Second, it rigorously evaluates all potential contamination risk factors and the overall state of control within the aseptic environment. Where are the weak spots? And crucially, it demonstrates that both aseptic operating practices and personnel procedures are appropriate and effective. Are people following the rules? Are the rules good enough? It's also a key tool used to qualify, requalify, or even disqualify personnel, ensuring everyone involved in the aseptic process is performing to the highest standards. Do the operators know what they're doing? Ultimately, it ensures strict compliance with GMP good manufacturing practice and all regulatory expectations for sterile products. It's a proof regulators demand.
That makes perfect sense. So how often do these critical APS runs happen, and how many are typically required for a new line or an established process?
The frequency and number of APS runs are carefully determined, always based on a thorough risk assessment of the aseptic process. It's not arbitrary. For a brand new facility or a novel process, regulatory expectations typically mandate a minimum of three consecutive successful APS runs. Got to prove it 3 times in a row. These are performed only after all other validation steps, including facility, equipment, decontamination systems, personnel training, room qualification, and the environmental monitoring program, are fully complete and proven. It's really one of the very last steps. For routine requalification of an already established line or process, semi-annual APS is the norm, twice a year typically. Any major changes to the facility, equipment, or the aseptic process also necessitate a comprehensive risk assessment to determine if and how many additional APS runs are required. If you change something big, you test again.
Now, this isn't just a simple run-of-the-mill test. It's about pushing the system to its absolute limits, identifying those worst-case scenarios where contamination is most likely to occur. How do you actually design these studies to truly stress the system?
You're absolutely right. It's about intelligent stress testing. A detailed risk assessment is the foundation for determining these worst-case scenarios. We consider a whole range of factors: the most challenging operating conditions, the maximum number and type of personnel interventions, like what if someone has to reach in, the most difficult container closure size or configuration, varying line speeds, and the largest batch sizes. What's the hardest thing this line has to do? To give you a clearer picture, we simulate a whole range of adverse conditions, everything from a brief power outage or critical filter failure to intentionally running the line at its absolute fastest speed, or even simulating changing tubing mid-run. The goal is to push the system to its breaking point under controlled conditions, to see how it recovers, maintains sterility, and ensures product integrity. Can it handle chaos? The duration of the media fill and the number of units filled are also absolutely critical for a meaningful simulation. Can't just run it for 5 minutes. The duration should accurately simulate either the longest routine fill a company performs or representative routine operations. Generally, for average production runs, filling 5,000 to 10,000 units is considered acceptable. However, for smaller production batches, less than 5,000 units, the APS batch should ideally equal the actual production size to be fully representative. You need enough units to find rare events. Conversely, for very large batches, those exceeding 10,000 units, the APS batch should also exceed 10,000 units, demonstrating the system's robustness at scale. BSM's qualification, for instance, is exceptionally rigorous, expecting up to 90,000 units in total during a single fill, covering a continuous 36 hours of fill time. 36 hours. That scale is what truly validates their ability to maintain sterility over extended real-world production cycles. That's a marathon.
You mentioned simulating adverse conditions. Can you give us some specific examples of the kinds of stresses the process is put under during these media fills for ampule and vial filling? Like, what do they actually do?
Certainly. These aren't just minor nudges, they're designed to replicate significant operational challenges. For ampulevial filling media, adverse conditions include scenarios like simulating a significant equipment stoppage, such as the filling machine being off for 60 minutes, maybe for unexpected maintenance, then immediately resuming and filling around 1,000 units. What happens after a long pause? Or simulating a critical environmental control failure, like the laminar airflow, LAF filtration, being off for 5 minutes and continuing to fill vials during that period, filling with bad air. Yikes. Even more extreme, they might simulate an air handling unit, AHU, power off for 5 minutes across all sterile and adjoining areas, then immediately resume filling, losing air pressure control. They can also simulate total power off for a brief period, then restart. Power blip simulation, running the line at its highest machine speed, such as 250 ampular vials per minute for 1,000 units, challenges its consistency at speed. Even seemingly minor events like a chain in product tubing mid-run are simulated. And critically, if an ampulevial sterilizing tunnel has a temporary fault, all units from the cooling zone are filled to see the impact. It's about finding out what happens when things go wrong and proving the system can handle it.
A critical element of media fills is understanding and rigorously simulating interventions. These are activities performed by personnel near the highly sensitive aseptic fill zone, and they must be kept to an absolute minimum due to the inherent risk of contamination. Every time someone reaches in, there's risk. A thorough risk assessment evaluates the contamination risk for each. Can you clarify the difference between the types of interventions?
Absolutely. Interventions are generally grouped into 2 categories. Inherent interventions are the routine, planned activities that are part of the normal aseptic process. Things like equipment setup, performing fill weight checks and making adjustments, recharging stoppers or caps, collecting environmental monitoring samples, or even scheduled shift changes. Stuff you have to do. Then there are corrective interventions, often referred to as non-routine. These are performed to correct an aseptic process during its execution, often due to an unforeseen issue, things going wrong. Examples include container breakage, dropped vial stopper jams, changing filling needles, clearing rejected units from the line, unscheduled maintenance work, or changing critical filters or tubing and pumps. Fixing problems on the fly. An approved list of these interventions, along with meticulously established procedures and thoroughly trained personnel, is absolutely essential to manage their inherent risk. You need a plan for the unexpected. For lyophilized products, where the drug is freeze-dried, the APS must capture the entire unique process. Loading partially stoppered units into the lyophilizer, the lyophilization process itself, and the final unloading, the whole freeze-drying dance. This can be simulated with shortened hold times, partial vacuum, and sterile air instead of nitrogen for venting to replicate worst-case conditions without actually freeze-drying for days. And of course, dynamic airflow visualization, commonly known as smoke studies, are crucial for aseptic filling lines. Gussy the air. These are performed during OQ and again during requalification. Their purpose is to visibly demonstrate unidirectional airflow in grade A zones, the most critical sterile areas during operation. Air should flow smoothly away from the product. Using a non-toxic smoke generator, teams video record smoke patterns in critical areas like fill points, stopper bowls, and conveyors under both static, nothing moving, and dynamic conditions, meaning with operators present and moving. How does movement affect airflow? The criteria are strict. The smoke must be uniformly swept away without any turbulence, reflux, or backflow, ensuring that air currents don't carry potential contaminants towards critical zones.
After all this rigorous filling with the growth media, what happens next? How do you actually assess for contamination? Do you just look at them?
After filling, the media-filled units undergo a crucial visual inspection pre-incubation. This step is to remove any non-integral units, meaning vials with cracks, pore crimps, or other physical defects that would compromise their integrity, but not cosmetic defects like a smudge on the outside. Got to make sure the container itself isn't leaky. The count of these integral units is carefully verified. How many good ones are going into the incubator? Then comes incubation. The units are inverted to ensure the media contacts all internal surfaces, maximizing the chance for any microbes to grow. Give the bugs a good soak. They're typically stored at 20-25 degrees C for 7 days, then 30-35 degrees C for another 7 days, or a single temperature for a minimum of 14 days, ensuring conditions are optimal for a wide-range microbial recovery. 2 different temperatures catch different types of bugs. Post-incubation, trained personnel visually inspect every single unit for microbial growth, indicated by cloudiness or visible particulates in the media. Is it cloudy? That's bad. Any non-integral units found at this stage after incubation are particularly alarming and warrant immediate, thorough investigation. Why did it break during incubation? The acceptance criteria for media fills are incredibly strict, with the ultimate target being 0 contaminated units. 0 is the goal. According to FDA guidance, for batch sizes less than 5,000 units, even 1 contaminated unit is cause for revalidation. Start over. For batches between 5,000 and 10,000 units, 1 contaminated unit triggers a thorough investigation and a possible repeat media fill. Find out why, maybe test again. 2 contaminated units means revalidation after investigation. Definitely start over. For very large batches, over 10,000 units, 1 contamination requires investigation. 2 again mean revalidation after investigation. The limits are tight. If any contamination does occur, a thorough investigation with root cause analysis and corrective preventive actions is absolutely mandatory. You can't just shrug it off. This includes species-level identification of the contaminant, what kind of bug is it, a holistic look at all systems and controls, environmental trend review, personnel activities and qualification, sanitization review, batch record and sterilization review, and tracing the contaminant's location to any interventions. It's full forensic analysis to find out how it got there.
This all sounds incredibly thorough, painstaking, and absolutely essential for patient safety. But let's be real, these processes are time-consuming, resource-intensive, and frankly, expensive. So what's the biggest challenge companies face when trying to implement all this, especially when they're juggling tight business goals and aggressive deadlines? Where does it usually break down?
That's precisely the crux of the matter, and life science consultant Devin Mack articulated it perfectly. He highlights the immense challenge of navigating the conflict between business goals and the deadlines attached to them, with everything needed to build a complete technical file. Business wants it fast, quality needs it right. Mack observes that companies often, in their rush to market, fail to set proper requirements at the outset, skip crucial documentation steps, and fundamentally misunderstand the essential must-haves for their technical files. They just want to get it done. They might even try to rely on predicate devices, similar products already on the market, only to discover later that their new product isn't quite similar enough to justify skipping tests, requiring them to go back to square one. That similar enough assumption can bite you.
So it sounds like it often comes down to a fundamental lack of understanding from the very beginning. As Devin Mack puts it, the crux of the matter is they don't really have a true understanding of what the requirements are. He also points to a historical shift where influence within companies moved from engineering teams, who traditionally prioritized thorough testing and validation, towards commercial teams, which sometimes led to justifications to avoid extensive testing in the name of speed to market, sales driving engineering decisions. Potentially risky,
That's right. For decades, engineering departments that championed exhaustive testing held significant sway, with VPs of engineering often being core to management teams. But then their influence waned. This led many companies to write justifications or rationale to avoid the amount of testing that they would have done 30 years ago, trying to rationalize shortcuts. However, new and stricter regulations, particularly like the European Medical Device Regulations, EU MDR, are now prompting a significant reevaluation and a return to rigor. Regulators are pushing back. As Mac notes, a lot of companies are pretty much faced with, okay, we actually do have to go back and test more than what we thought. Reality check time. This crucial shift underscores that real data, comprehensive control over the product realization process, and verifiable proof of quality are once again becoming non-negotiable. You need the proof. It's a painful but necessary recalibration for some.
So if companies are finding themselves in this bind, needing to go back and retest, what are the practical solutions and advice from experts like Dev and Mac to prevent these costly, time-consuming missteps from happening in the first place? It sounds like it all starts right at the foundation. How do you avoid the rework?
Mac's advice is clear, practical, and foundational. First, he urges companies to challenge any assumptions made early in the product realization process, especially those that rationalize omitting or deferring critical activities. Question everything early. Second, devote ample time upfront to thoroughly laying out the full set of requirements with essential input from every impacted department, not just R&D or marketing talking amongst themselves. Get everyone in the room. Third, acknowledge that few decisions can ever be responsibly made in a silo, especially early on. Manufacturing teams, for example, should be intimately involved from the very start, contributing their expertise and foresight. Ask the people who have to build it. The ideal, he states, is meticulous cross-functional planning, where requirements perfectly align with design inputs and outputs, involving everyone in the conversation from the earliest conceptual stages. It's about building quality in, not inspecting it out at the end.
So it truly comes down to foundational planning, ensuring that quality isn't an afterthought or a last-minute inspection, but woven into the very fabric of the product's journey right from its conception. This brings us to a powerful concept known as quality by design, QBD. It sounds like more than just a buzzword.
Exactly. Quality by design is the overarching philosophy that truly drives the effectiveness of all this qualification and validation we've discussed. It's a mindset. It means consistently approaching every step of product development and manufacturing in ways that have proven effective time and again, embedding quality into every decision. A key aspect is deeply understanding your customers, who could be actual patients or healthcare providers, by identifying their human factor requirements and translating those into actionable design inputs. What do users really need? As Devin Mack explains, how do you know you've met success? It's by doing things in a proven way, and a big part of that is understanding your customer. Tools like value chain analysis, Kano diagrams, and customer surveys help interpret these often qualitative requirements at a precise engineering level, turning fuzzy needs into concrete specs. Success, then, is truly measured not just by the product's technical specifications, but by meeting these explicit and implicit customer requirements and the clear, measurable acceptance criteria outlined in every IQ, OQ, and PQ. If those criteria are vague, companies will inevitably struggle to define what success even looks like, and more importantly, struggle to consistently achieve it. You need clear goalposts.
Given the intense, cyclical, and highly specialized nature of this work, constantly needing to hire and retain highly skilled in-house experts for equipment qualification and validation, it isn't always feasible or cost-efficient for most organizations, especially smaller or mid-sized ones. It sounds expensive to keep that expertise on staff full-time. So what's an effective model that many companies are turning to for managing this critical but fluctuating demand?
Many life science organizations are finding a smart, adaptable solution in outsourcing this specialized work to specialized resourcing firms, often through flexible contract staffing or staff augmentation models. Bring in the experts when you need them. This approach offers significant advantages. It provides crucial access to highly qualified and experienced personnel in an incredibly competitive labor environment. Good validation people are hard to find. It frees up valuable time for internal teams to focus on their core competencies, and it dramatically reduces the substantial costs associated with recruitment, onboarding, and long-term employment. Less overhead. Furthermore, it allows companies to easily adjust to the cyclical or project-based demands of validation work, scaling up or down as needed. Need 5 people for 6 months. Okay. Need 1 person next year. Okay. It also has the added benefit of infusing new skills and diverse experiences into the team without the long-term commitment of a full-time hire. Fresh eyes can be good, It's a highly adaptable strategy for achieving and maintaining quality by design without building massive, often underutilized internal teams. Hashtag tag outro.
Today, we've journeyed deep into the intricate world of equipment qualification and process validation. We started with the foundational why and the legal mandates, moved through the essential steps of factory acceptance testing, site acceptance testing, installation qualification, operational qualification, and performance qualification. Phew. We then saw how this all plays out in the incredible complexity of a multi-vendor sterile filling line, and culminated with the ultimate challenge of aseptic process simulation, or media fills, meticulously designed to simulate worst-case scenarios and guarantee sterility. What stands out to you, I hope, is how deeply intertwined engineering, quality assurance, and even strategic business planning are ensuring the safety and efficacy of the products you might one day rely on. It's a testament to meticulous planning, rigorous testing, an unwavering commitment to quality by design, and continuous vigilance that often goes entirely unseen, but which is absolutely essential for public health. So much happens behind the scenes.
This raises an important question for all of us. If validation is so critical, it's clearly not just a one-time event, is it? The real challenge and the true measure of a robust system lies in maintaining that validated state, constantly monitoring, adapting, and re-verifying as processes evolve, as technology changes, and as new risks emerge. It's a continuous cycle of vigilance and improvement that truly never ends. You're never really done.
So after all this rigorous testing, documentation, and continuous vigilance, what if a company's product, despite passing all these hurdles, still encounters an unforeseen issue in the real world? How does the very framework of IQ, OQ, and PQ, this incredibly detailed foundational understanding of their equipment and processes, empower them not just to react, but to rapidly predict, prevent, and continuously refine their processes for an ever-safer future? It's a fascinating thought to consider and a powerful reminder that true quality is an ongoing journey, not a static destination.